Alright, so let's see where we are. So we definitely drew our chloroplast. We are skipping this emergent. I'll mention it, we'll see. Where can you find them? We drew that, mesophyll cells. Then we took this figure that was here and we labeled it. So that's done. We have included the equations for photosynthesis. We've done that. We talked about water being split, but this is now getting into the light reactions, and so this is what we're going to be doing. So we're going to be doing the water being split in our notes, source of electrons. We're going to summarize the light reactions. We'll remind ourselves where we've written chemiosmosis before, but we'll write it again. And then this will be the last video that we'll talk about Calvin, and we'll go over here with, well, hey, what if this part changes and we don't have this particular molecule? What does that affect? And then this whole part starts a new piece. So we'll finish the properties of light. We've summarized the light reaction, what we're doing today, which includes cyclic photophosphorylation. We've already defined all of these before. I may leave those for you to go back and do. We're going to talk about the difference with oxidative phosphorylation and photophosphorylation. Easy peasy. And then spatial organization as far as in the chloroplasts. This is what they're asking you is about the gradient. So we'll talk about that. Okay, then you're going to have things about different plant types that have developed ways of dealing with harsh environments. So you've got C4 plants, which is what you're used to, C3 plants, and CAM. And honestly, it's really not an easy concept. It takes a lot of time to go over, and they rarely have a question. So Godsey and I decided we're not going to talk about those. You're going to want to, so what we'll do is we'll assign some reading for you to do in case there's a question or two on your test. And then we'll talk about inhibiting one part of photosynthesis and if it affects the other part. And we have yet to talk about these two concepts. Photorespiration. I mentioned this when I talked to you about Rubisco. And I think right through here we'll assign some type of reading in case you have a question on those. Okay, so pretty much we're going to be done with notes here in the next couple of videos. So here's where we are. Ignore this is where I was just playing around with what I wanted to draw and it was way too messy. This is much better. The room, desk, it's not a desk. So this is where we left. And we made sure that we looked at the chloroplast. I'm going to go back in and add my detail here. That that's the outer layer. This is the inner membrane. Just making sure that my details line up for everything. And then what we're really going to do with this whole conversation is that follow the electrons. Again. Because whether you're making or breaking bonds, the most important thing about it all would be the part that's the most dangerous if it's loose, which would be your electrons. So we'll follow the electrons. So we're going to have two paths where that we tell the story. We're going to have linear, we talked about it yesterday, but we're going to actually write it down, what goes where, and what goes through where, and then we'll do cyclic, and then we'll talk about the gradient, and that'll help us get to that little question we had. Okay, so follow the electrons. So when you're talking about linear. Linear means that you don't revisit any part of the pathway. Everything travels through one time. Like the electron does not revisit and get picked up by any molecule or go through any part of the process more than once. So all of this is called photophosphorylation. Couldn't say that and write it all at the same time. And all it means is you're going to phosphorylate, which means adding a phosphate group to something. And in this case, it's going to be making ATP using light energy. Okay, so you're making ATP using light energy. So the first type is linear. So your electrons are going to start in water. And that's what we're doing. We're following the electrons. So you're going to have the electrons are in water. As the light energy hits photosystem 2, its own electrons, which, you know what I think I'm going to do? I want to show you what that pigment molecule is. I don't know if you've ever gone back and watched like the old Disney movies and the ones that they hand drew every single frame. They're beautiful, but it's crazy what we'll get used to with this whole digital animation stuff. And this is a really old 80s, maybe, video from TV Ontario. And so what they're doing is they're explaining, and hardly any place else shows it, so we're going to watch this one. But they're showing what happens when light energy hits the pigments and the way that the electrons pop out of the pigment and are replaced by water. But I want to start at this part because what you see is right through here in this middle where it's green, this is where you have chlorophyll A. It's in the photosystem 2. It's at the very beginning. But you have these look like little Pac-Man pieces that have different colors. And one of our questions asks us, why do you have these other colors of pigment? So I don't know if you know why leaves change color. And what happens is that the primary pigment is chlorophyll A, which is green. And so as the days become shorter and the temperature changes to where there's a difference in temperature over the time during the day, in other words, you're going into fall, the leaf begins to shut down making, and it makes less of the chlorophyll A green pigment. And so these carotenoids, which are the more yellowy, orange, reddish type pigments, they're always there, but they're called accessory pigments, and so they're in less amount. And so you don't really see them. But as the green stops being made, and so you're going into fall, the tree continues to make the accessory pigments, the carotenoids, and that's why the leaves then appear to be, bright red, orange, all those yellow, gorgeous colors. And then they shut that pigment system down, and then the leaves drop. So that's what they're showing here, is that you've got not only the main pigment that is going to be excited by light, just a certain wavelength of light, which is chlorophyll, but these accessory pigments as well. And what they do is they're able to pick up the wavelengths from a different part of the spectrum, and we'll get more into that in the next little video. And then they channel that over to and add on to the energy that's going to be used to excite the electrons. Okay, so they're going to walk you through how this works, and let's pay attention to the reasoning behind the structure of the actual pigment molecule and how come it can donate electrons when it gets excited by light.
Because of the way they trap and redirect the energy of incoming light, these pigments are called antenna pigments. antenna on a rooftop intercepts television signals and passes their energy onto your set, while antenna pigments intercept light waves and pass their energy on to chlorophyll A. Well, you receive a better picture on your television set. Chlorophyll A receives more of the sun's light energy to carry out photosynthesis. Even though chlorophyll B has nearly the same molecular structure as chlorophyll A, it actually functions as an antenna pigment. Despite the differences in function of chlorophyll A and the antenna pigment, they all share an important feature: alternating single and double bonds. These alternating bonds can be easily seen here with chlorophyll, and here with the beta-carotene molecule. The double bonds represent electrons less tightly bound to the molecule. And because these are weak bonds, it takes less energy to jolt the electrons out of their orbits. In photosynthesis, that jolt is provided by light energy. Light, which exhibits properties of both waves and particles, consists of discrete bundles of energy called photons. Since each pigment molecule has very specific energy requirements, it only absorbs those wavelengths of light and those photons which supply the necessary pressure. As we have seen, the antenna pigment, the chlorophyll A and chlorophyll B, absorb photons with energy other than those accepted with chlorophyll A. The antenna pigment then passes the energy of the photons on to chlorophyll A in buckets of gauge. To do this effectively, the pigments are grouped together successfully to form an action set called photosystem.
Okay, so, I mean, I know that's really the best animation, but you can see where that chlorophyll A is going to use a certain wavelength, and it's going to have, those double bonds are much, much weaker, and so it's easier to jolt electrons out of their orbit from the light energy striking them. But then you get this handoff of electrons from these accessory pigments as well. So I wanted you to see how that works, because there's so many things that are just misspeaks. So when people are walking through how this goes. So the electrons are going to come out of photosystem 2, and they're replaced by water. So considering that, if they came out of photosystem 2 and you didn't replace them, then that would be a whole different story. But since the replacement electrons, the ones that continue to flow, come from water, we're going to start with the electrons coming from water. These electrons then are going to move through photosystem 2, and it's all of that ability to harness the light energy to cause those electrons to become excited, high energy, and then they're going to be picked up by Q. So I'm going to come over here and put that the path goes from water to Q, and then the next electron acceptor is C, followed by, which we wrote this yesterday, we wrote ferredoxin, but we'll just put, it goes to ferredoxin. This is where we get the decision pattern of, do we continue going straight to making NADPH? If so, the electrons go from ferredoxin to NADP. So that's the pathway. As they're moving, so as the electrons move from water to Q, they're going to go through excitement in photosystem 2. As the electrons move, now they're high energy. We'll come under here and write whether they're high energy or low energy, which really they're low and the rest of the time they're high. As they go from Q over to C, they're passed through an electron transport chain and all that. That's like a fancy thing, and people say that and they don't have a clue what they're saying. Electron transport chain just means you are passing electrons between different electron acceptors. So you're handing it off just like you hand off, what is it called, baton in a relay race? That's the exact same thing, except the baton would be the electrons. So you're you're transporting the electrons. And so it goes from Q to C, and it goes through, as it drops in energy, that energy is collected by this proton pump on the way to C, because we drop in energy, the electron does, because Q doesn't hold the electron as tight as C does. So we're going to come over here, and we're going to put that the electron's passage through a proton pump to C. Now they're low energy. So I misspoke when I said that from then on they're high energy. It's going to be low, high, low. And then we're going to ramp up with light energy again by using the photosystem, this time different wavelength. And we now have high energy electrons with ferredoxin, and ferredoxin will passage those electrons to NADP, and it uses this enzyme called NADP reductase. So I'm not going to write that up there because it's just going to hand them off. So as far as the energy level of the electrons, when they're in water, they're low. Okay? Oxygen holds them really tight. Water's not a big molecule. It doesn't share the electrons equally with hydrogens. That's what makes the whole molecule polar. It's a very low, stable energy bond with the electrons. Then you ramp up with photosystem 2 light energy, and when they go to Q, now they're high energy. And instead of just handing them off to NADP and being done with it, we're going to use the energy in those electrons. We're going to passage them down to C. C's going to hold them tighter than Q, and so that's going to drop the energy. And we use this drop in energy, which we're going to come over here and draw this, to run the electron transport chain. Now we've got to re-energize that electron, which we do with light, and we're back to a high energy electron that's handed to NADP, which is reduced, so it becomes NADPH, and it carries that high energy electron pair to the Calvin cycle in order to put some carbon dioxides together. So let's draw what this looks like. So we're going to go and start down here with low, and we're just going to do a series of steps. So we're increasing the energy of the electron, and now it's high. This increase is what we've used in order to increase it. We used light. Now, one thing I will tell you, there are some just tricks to make sure that you get your points. And light needs to be accompanied by energy, light energy. Don't ever say kinetic. Don't ever just say energy. If it's energy of movement, say kinetic energy. So you want to put it all together. So light energy is what's going to change the energy level of the electron. So now we've gone, our electron's gone step up to high energy, and then we're going to reduce the energy of the electron back down because we've passaged it from an electron acceptor that holds it less tight, lower in electronegativity, to something that holds it tighter. And so this is going to be where we use that drop in through an electron transport chain. So we're back to being low again, and we're going to step up the energy again, and we're going to use light energy. And this is in photosystem 1. And now we've got high energy electrons that we do, that we provide to Calvin in order to make the precursor molecules for glucose. So I hope visually that helps you. So I'm going to circle linear. Boy, my pen's running out. I go through a lot of pens. And so we understand that a whole lot of this is very similar. So I'm going to do cyclic, well, you can't see that, in red. And so this is cyclic photophosphorylation, and I'm going to follow this whole thing, and I'm just going to overlay it with red. So we still start everything off the same. You've got photosystem 2, all of the, which we showed, those double alternating double and single bonds. Light hits it, you pop electrons out of one of the double bonds. They're replaced by splitting water. So they still start with water, go through the photosystem 2 to Q. Same thing. So now they're high energy. Then we're going to passage those through the electron transport chain, which means you're going to pump hydrogen ions by using that drop in energy to power your pump. Now your energy of your electrons is low again. So we're going to re-excite them with a different wavelength of light. So now we're back up to high energy. And here at ferredoxin, depending upon the conditions in the cell, if there's an insufficient amount of ATP for Calvin to work, as I told you Calvin's a pig, then ferredoxin will send the electrons back through the electron transport chain. So it goes back here, and you would have this reusing, because it's high energy of the electron, use that energy to go through the electron transport chain, create a greater gradient, that's like interesting, a larger gradient, because you're pumping more, energy's low, re-excite, ferredoxin would resend, and you just continue with this short circuit right here for cyclic. So at ferredoxin, the electron pair is sent back to the electron transport chain to create more hydrogen ions pumped. And we'll put therefore it increases the gradient. And what that does is, this is that whole relationship thing I told you. More hydrogen ions, more of a gradient, more ATP that can be made. And that's what cyclic is. Okay, so I don't know that there's really anything else that's important other than as ATP is needed, ATP synthase is opened, and so you exploit this hydrogen ion gradient that's high in the lumen of the thylakoid that's been put there by pumping hydrogen ions against the gradient, using that drop in electron energy to power that pump. And you've got all these hydrogen ions, and you just let them flow according to their natural gradient. So they're going to move based on that repulsion, that chemical repulsion they have, because they're crammed in here, a bunch of positive charges, and also just the natural entropy of moving from this ordered system, which is high here and low here, to random, which would be the same on both sides. That's what entropy is all about. Things move naturally toward disorder. Having high here and low here as far as concentration of hydrogen ions is ordered. And so through entropy, diffusion occurs, and you're going to have high hydrogen ion concentration moving through ATP synthase. And this is an actual motor. It spins. It has what's called a catalytic knob. So it grabs that energy from the revolving door and changes mechanical energy of spinning, because these hydrogens are going through that revolving door right here. They come in, they spin, and then as they go out, that mechanical energy is transferred through that catalytic knob to provide enough energy to create this ATP. Okay, so I hope that makes sense for the notes for all of the pieces there with the light reactions. We used to go crazy and talk about what the nanometers of light are, and I may come back and add that when we get to the part where we have to talk about what light's made out of. But for right now, I'm happy with this one. I think it's enough as long as it's clear.